B cells are part of the humoral immune response. Activated B cells secrete antibodies, antigen-binding proteins that circulate in the blood and lymph. These fluids used to be known as humors, which is how the humoral response was named. B cells develop in the bone marrow. The B in B cells originally described where they developed in birds, an organ called the bursa of Fabricius. But since B cells develop in the bone marrow in all other vertebrates, the B can also help you remember bone marrow. B cells are antigen-presenting cells, so they express MHC2 molecules on their surface. In addition, membrane-bound antibodies on the surface of B cells act as receptors for antigens. When an antigen binds to a membrane-bound antibody, it's taken into the cell by endocytosis and digested. In a process similar to the presentation of antigens by macrophages, the antigen fragment is expressed on the surface via an MHC2 molecule. Since each B cell has membrane-bound antibodies for only one specific antigen, the same antigen is presented on all of the MHC2 molecules of the cell. A helper T cell with antigen receptors for the presented antigen will interact with the B cell. The helper T cell secretes interleukin-2, which activates the B cell. The activated B cell divides many times to produce memory B cells and effector B cells. The memory cells respond if the antigen enters the body again. The effector cells secrete antibodies to fight the infection. An effector cell can secrete about 2,000 antibody molecules per second.